presentation, and I'm very happy to share some work I'm very excited about. And so this is to do with population dynamics in moving environments. And let me just see if I can get my slides to work. So many populations grow and disperse in moving environments. And a kind of a classical case is populations in rivers that are subject to downstream flow. And actually, there was a paradox that has been asked since about the 1950s, which asks how river populations can actually persist spatially without being washed out when they're subject to continuous downstream flow. And a mathematically related problem is related to climate change. It asks how weather populations subject to shifting environmental conditions will be able to keep up with shifting temperature isoclines. And mathematically, these two problems are very closely related. If you sort of go to a moving coordinate system, that's describing the movement of the temperature isoclines. So I'm going to draw on some of that today. But there's more questions here. One question is, suppose populations do keep up with shifting environmental conditions that are induced by temperature changes. It's possible that the structure of the populations will change, for example, the genetical structure. And so in this talk, I'm going to focus on coupling genetics to population growth models to look at the consequences of climate change. But I'm going to do it in the context of populations in moving environments and draw on that kind of mathematical theory. But if we just look at climate change predictions, of course, this will be quite familiar, these kinds of graphs. This is from the International Panel on Climate Change. And this is the global mean temperature shown on the vertical axis and the date on the horizontal axis. And of course, the projections depend on our human actions. And there's a business as usual scenario, which is in the red, and the strong mitigation scenario, which is in the blue. And presumably, the actual temperatures will be somewhere in between. And so there is this, of course, increase in temperatures. And these warming temperatures cause shifts in temperature isoclines, either towards the poles or towards higher altitudes. And species can respond either by migrating or by adapting, and migrating either up mountains or towards the poles. And so in our mathematical model, we'll have a patch of habitat, which we call good habitat. And that habitat will be shifting at some speed, c. And in this case, it's shifting to the right, but we can think about that as being the north. And so the climate envelope is a patch of good habitat surrounded by bad habitat. And then the speed at which this is shifting, we're thinking of as c. Okay, so the International Panel on Climate Change has sort of wondered about whether species can keep up with climate change speed. And so this is a graph from their 2014 report. And on the left-hand side is the maximum speed at which species can move or have been observed to move through historical data. And we see they're different for different species, like some mammals have spread into new habitats very quickly. And then we see trees are on this scale here. And this is kilometers per decade. And then on the right-hand axis is the actual predictions for average climate velocity in terms of these temperature isoclines. And of course, they're also highly variable depending on how far north you are and the mitigation scenario and so on and so forth. So if these dotted lines fall above the bars for the animals, then the idea is that then they would likely be unable to keep up in terms of historical record with their migration as the habitat shifts. But there's more to the story, like I mentioned before, and this is to do with genetic diversity. And so it's well known that things called founder effects occur when the migration into a new habitat is just by a few individuals. And if just only a few individuals shift into this new habitat and then grow and disperse, then there could be a reduction in genetic diversity. And this idea has been around for a long time. I think one of the earliest references was by Ernst Mayr in the 1940s. And so we might ask, even if populations keep up with climate change, are they able to maintain sufficient genetic diversity at the leading edge of migration? And so from the mathematical perspective, we may be able to calculate an intrinsic spreading speed for a species due to combined effects of growth and dispersal. And this could come from a mechanistic model for growth and dispersal. And then we could simply ask, is that speed greater or less than the prediction for climate change? And that would be taking the approach that the IPCC report took. But there is a subtlety here with respect to the size of the climate envelope. And so perhaps a large envelope will allow for a different amount of survival than a small envelope. Some species like butterflies actually have quite small spatial envelopes in which their host species are shifting. And so perhaps if that envelope got very small, then it would affect the survival or the population persistence. But then if the population does persist, how does the genetic diversity change over time? Are there significant founder effects? Or if there are founder effects, when do those occur? And so this is something we're going to look at today. And then finally, you can do a sort of a comparison, because this issue of a founder effect has already been sort of observed historically in terms of range expansions into new environments or biological invasions. And so we might ask ourselves, how does the range shifts due to climate change, how are they different from just simple expansion of a population into a homogeneous environment that would come from introducing a few individuals and letting them spread? And so this is my outline. I'll start the next bit by talking about the intrinsic spreading speed, and this draws on some beautiful classical theory that goes back to the 1930s for populations. And then I'll talk about how we calculate this speed. And there's a difference between pushed and pulled waves depending on their growth dynamics. And this actually plays a very important role in terms of the genetic diversity dynamics. And then in part 4, I'll talk about tracking neutral fractions for spreading populations. So I'll look at a particular kind of genetic model that assumes that the gene fractions are neutral in that they don't confer a selective advantage or disadvantage, but they're simply markers that are getting rearranged by this population spread. And then I'll talk about a moving environment. And so this would be one of these habitat patches shifting at a given speed. And then when I do that, I'll first start talking about the population persistence question during range shift. And then I'll move to tracking neutral fractions in these habitat patches. And then finally, because things by that time will be kind of complicated, I'll try to simplify things down into kind of simpler ways that we can measure genetic diversity for these kinds of questions that are easier to communicate than solutions to partial differential equations. And I'll end with conclusions. Okay, so the model for the spread of an advantageous gene into a new environment was proposed by Fisher in the 1930s. And this has actually been a model that's been used for population growth as well. And so the idea here is we have logistic growth and diffusion. And the logistic growth function is shown at the bottom. And so we have a carrying capacity, which has been set to be 1 here. And I've noted the units for growth and diffusion, as we'll come back to these later. One of the early researchers, research groups looking at this, Kolmogorov, Petrovsky, and Piskunov in the 1930s, showed that a step function of initial data converges to a wave that has a speed 2 times the square root of r times d. And this is a very kind of classical result in mathematical biology. It turns out that that particular initial data isn't necessarily the most interesting, and slightly more interesting cases where there's initial data which is on a compact, non-zero on a compact set, U0, and 0 elsewhere. And this would correspond perhaps to a biological invasion where individuals are introduced just on a local region. And Aronson and Weinberger had these beautiful papers in the 1970s that looked at this case. And so here, this compact initial data converges to a wave expanding in 2 directions at speed c star. And the idea behind their proof to show this was to create these sub and super solutions, one that lay below the initial distribution and one that lay above. And both of these solutions end off moving at the speed c star. And then they invoke an ordering principle that comes from the comparison theorem that says that the true solution has to remain sandwiched between the upper and lower solution. They can't pass through each other. And so its speed has to asymptotically be a c star as well. And so this is beautiful mathematical theory that, I mean, of course, is quite old, but even older is work by a chemist called Luther. And he was looking at these spatially distributed chemical reactions. And he argued that the rate of the chemical reaction spreading across the substrate was proportional to the square root of r times d. And his argument was just simply based on dimensional analysis. So if you look at r, it's got units 1 over time. D has got units space squared over time. And if you multiply them and take the square root, you get something with the right units. And that preceded the other work. Of course, he didn't know the ratio of the right constant to put in there. Okay, so this is going to be useful as we start to look at the intrinsic spreading speed for populations. And you might ask, how do we calculate this speed? And so at the top, we have a nonlinear model. And of course, we don't really know how to solve nonlinear problems usually, but we're very good at solving linear problems. And so wouldn't it be nice if we could just solve the linear problem and use that to say something about the nonlinear problem? And that's the case here for Fisher's equation. And so if we linearize about the leading edge of the wave, i.e. u is equal to 0, then and we solve this linear problem, we can write down a solution to that, and that solution will asymptotically spread at the speed c star that is given for Fisher's equation. And so we say that in this case, the spread rate is linearly determined. And so we can sort of ignore those nonlinear higher order terms. This is not always the case though. So Fisher's equation is sort of shown on the left, where the linearization lies above the nonlinear function. But on the right-hand side, we see a case where the linearization about 0 doesn't lie above the nonlinear function everywhere. And so it turns out that a sufficient condition for linear determinacy is that we have something that looks like the picture on the left, where the linearization lies above the nonlinear function. And in that case, we say that the wave is pulled by the leading edge of the invasion, because this linearization is describing what happens at the leading edge, way out at the edge of where the population is spreading. So you might ask yourself, what happens in the case on the right-hand side? And so in that case, the rate of spread may not be linearly determined. And so there's this idea of a pushed wave. And so with a pushed wave, populations at intermediate density grow fastest, and they spill over into the leading area, and then they cause the spread of the invasion. And this idea of a pushed wave was introduced by Hadler and Rote in the 70s. And often these pushed waves are seen when there is an Allee effect. And so ecologically, an Allee effect means that populations at lower densities have a reduced per capita growth rate because they're social feeders or they need to reproduce sexually. And so the highest per capita growth rate is not at the lowest densities, it's at intermediate densities. And so here, there's the idea that you might have a weak Allee effect, which would be something like the function shown currently on the right here, where there's still only two zeros, one at 0 and one at 1. And a strong alley effect is the bistable equation. And it's the case where rho lies between 0 and 1, where there's actually 3 roots and the population growth rate is negative for sufficiently small population sizes. And I'm going to be using this a bit later. So what's really fascinating is that this theory has been around for a long time, and people have tried to connect it to field observations of population spreading. But very recently, there's been some work by Gandhi et al., which is very beautiful. So they manipulated yeast populations on Petri dishes by changing the growth medium. And so the idea is that glucose is like high octane fuel and it promotes individual growth, whereas sucrose is a more complex sugar, and the yeast have to break down the sugar before they can use it. And so they become cooperative feeders when they're fed sucrose. And so what this group did was they analyzed the wave speed and the shape of the wave that came from glucose and sucrose. And they found that with glucose, we have a pooled expansion where the individual growth at the leading edge was the highest. And with sucrose, there's a pushed expansion where growth at these intermediate densities spilled over and caused the wave to move forward. And so this is quite recent work, and it's a very it can be repeated easily in the lab, which is nice. It took a long time from the original theory to see this kind of work. So it's also a nice example of where theory has suggested an experiment that has been then used to test the theory. So let's think about tracking neutral fractions for a spreading population. And so now we're moving to the genetics a little bit. And so we consider the dynamics of these fractions in the wavefront, and we're going to move to a traveling wave coordinate system, which is shifting at wave speed, c, across the domain. And we're going to break it into these fractions. And the fractions differ only by their position in the wave and their allele or label. And so we see a scenario that looks a little bit like this. And so this is a wave moving at speed c to the right, and gray is one type of genetic fraction and red is another, and so on. And so in the model, we're not going to confer any different genetic advantage or disadvantage to the different colors. They're just these neutral markers. And so it's sort of a special case. But it turns out there's a lot we can do with this case. And so we're breaking down the wave into these neutral fractions here initially. And then we ask how they change over time as the front moves forward. Does it keep that shape, or do they mix up, or what do we see? And so these changes are governed by what are called inside dynamics. And Roch and Garnier, this French group of PDE analysts, looked at this case and developed some theory for it, which is very beautiful. And so one thing we can do is start by just writing down the dynamics of these fractions. And so this is the ith fraction we see here, and then there's diffusion, and then there's growth. And now we've broken our growth into per capita growth term G, which is given below as F of U over U. And then this is the neutral fraction density, V superscript I. And so this is sort of interesting. This equation is linear in V superscript I, even though it involves the total density of fractions U here. And so were we to add up all of these equations, we would get back our original Fisher type model, or nonlinear reaction diffusion model. And so I'm just going to show some numerical solutions of what happens here, and then we'll talk about some mathematical results. And so here on panel A are the initial conditions for a spreading population. And so it's just a top hat distribution. And you can see there's very distinct slices like a loaf of bread, where gray is on the right and then comes red and orange and so forth. And if we just have the Fisher equation case, which is shown in panel B, it's only the very leading edge that is asymptotically seen as time goes to infinity. These other fractions get left behind. And the idea there is because it's a pulled wave, it's just those individuals at low density that are spreading and pulling the wave along. By way of contrast, if we have an alley effect, this is the case where the spreading speed is not linearly determined, then if we look at the leading edge of the wave, we're seeing all the different colors here. There's gray and red and orange and so forth. And when the alley effect becomes stronger, we see even more diversity in the leading edge of the wave here. So B would be a case with limited diversity at the leading edge of the wave, whereas C and D would have higher diversity. And so, so what Garnier et al., Jimmy Garnier and Rock showed in about 2012 was that the founder effect was most pronounced with these pulled waves and least pronounced when there's a strong alley effect. And that's, in some way, the alley effect is actually helping preserve neutral genetic diversity in the spreading wavefront. And so I've just rewritten those points again on this slide. And they have a theorem about this, which is quite easy to read in some sense. So in the pulled case, which they call the KPP case, they say basically if the initial density of a neutral fraction converges to 0 faster than the traveling wavefront, then asymptotically, as time goes to infinity, you don't see it in the leading edge of the wave. That's what the first case says. Whereas if there's an alley effect, then the density converges to a proportion of the leading edge of the wave. And so what it's saying is that in actual fact, if we just zoom in at the leading edge of this wave, each of these profiles is going to take the same profile as the total density, and each of them will have a different proportion of that total density. And so you see all the components. And so that is that each of these converge to some proportion P of this traveling wavefront uniformly as time goes to infinity. And it turns out it's a sort of a beautiful result because that proportion can actually be computed explicitly. And there's a formula for that. And the fact that this theorem works sort of relies on the fact that these dynamics for the neutral fractions are actually linear equations. And so we can do lots of things with them. And so what does this say here? Well, this is your initial distribution of a particular neutral fraction. And this is the traveling wave front, okay? And so we're taking the product of these two things, and then we're putting a weighting function in here. And the weighting function is gonna emphasize things that are further to the right, have a larger value of X. And the weighting term is C over D. And so, so the idea here is, if we go back to the pictures, that, for example, if we look at case C, we're going to get C is further to the right initially, and so that weighting term will be larger. And so we're going to see more red in the wavefront than we are going to be seeing dark gray, for example. And but what Rock and Garnier did was they actually had a formula for these different proportions for the different colors at the leading edge of the wave. And that's the formula given right there. And so just very recently, the same group that looked at these pulled and pushed waves using yeast actually did a very beautiful experiment using genetic markers on the same types of populations. And so what they found they could do is they could put what was essentially a neutral genetic marker on a yeast population that would fluoresce. And so it's a fluorescent marker, and so it could be tracked over time. And so they could run these experiments for pushed and pulled waves, and then they could manipulate the substrate by going from glucose to sucrose, so pulled to pushed, and then they could see how the genetic diversity of the leading edge of the wave would change. And so this is, again, the picture that we saw previously on the left, where we have a pulled expansion for glucose and a pushed expansion for sucrose. And the idea from the theory is that the pulled expansion should have reduced genetic diversity, because it's only those individuals at the leading edge of the wave that are going to contribute as time goes to infinity, whereas the pushed expansion should have higher genetic diversity. And so on the right is the outcome, or is a little summary that I did of the outcome of the experiments by Gandhi et al. And so they have a quantity they call pushedness, and the red wave has got a pushedness of around 2, and the green wave has a pushedness of about 1. And so low pushedness. And on the vertical axis is a measure of genetic diversity, which they call effective population size. And so the population that is pushed more has higher diversity, and that the one that's pulled has the lower diversity. So again, this was an experiment that was driven exactly by the mathematical theory and other kind of computational verifications of that theory. And it's a sort of a beautiful example of how they can do these experiments in a replicable way. Okay, so this is kind of the theory without climate change or a moving environment. So let's shift gears a little bit and now talk about including a moving environment. And the first thing we can do is just think about our original model, but now go to something with some advection velocity. And so A is the advection velocity, and it's just shifting things to the right at velocity A. And so we can sort of imagine for now that this is an infinite river that we're looking at. And there'll be an upstream direction and there'll be a downstream direction. And the flow is shifting everything downstream. And it's fairly straightforward to show that then the speed in the upstream direction is the Fisher speed minus the shifting speed for the advection. And the downstream speed is Fisher speed plus A. And so if we were to plot this with the horizontal axis being the advection speed in the river and the vertical axis being the up and downstream spreading speeds, in the downstream, the advection is always going to speed things up, and the higher the advection speed, the faster it'll spread downstream. In the upstream direction, it's a little bit different because we have a point here exactly when the advection speed is the same as C star, where the upstream spread will stall and it'll no longer spread upstream. And if the advection speed gets larger than that, then it'll actually, the upstream spreading speed will be negative and it'll be shifting downstream. And that would be like a washout case in the river where populations couldn't persist. So we can think about this a different way though, and I want to look at it both ways and kind of compare. And we can add in this idea that there's a patch. And so here we have a patch of size L within which there's Fisher growth, and outside of which there's exponential decay of populations. So outside of this patch, we think of it as being a hostile environment where populations only go there to die. Inside the patch, we'd have logistic growth. And so now this is given in the F term. So there's a spatial heterogeneity in the growth term. And so here, just to remind ourselves, A is the advection speed. L is the length of the good patch that we see in this formula for G. D is the death rate in unsuitable habitats. And R is the familiar per capita growth rate in the good habitat. And now big D is the diffusion coefficient. And so we can sort of look at this problem and think about whether we could have a non-trivial solution to this problem. And so we might say there could be a zero solution, and we want to have a solution that persists. So you might ask yourself, how large a patch do we need, L star, for a population to persist in the system. And so the idea here is to look for the existence of a non-trivial steady state solution. So we've got the same equation, but now we're looking for something that doesn't change in time. So the du/dt term is gone. And so we'd like to have a solution to this which is non-zero. And whether or not such a solution exists depends critically on the size of the patch L. And we would imagine maybe with small patches, we wouldn't have such a solution, but for large patches, we could. One little trick that comes here is to translate this problem on an infinite domain to a problem on a finite domain. And this is through these mixed boundary conditions. And this is using a trick by Don Ludwig and other people from the 70s, where it's possible to actually make this conversion. And so the idea is here that we're now looking for the existence of the steady state solution on a finite domain that goes from 0 to L with these boundary conditions. And the boundary conditions involve the death rate, the diffusion coefficient, and the advection speed here. And so this makes the mathematics a little bit easier. We don't have to look at it on the infinite domain. And then we ask ourselves now, how large a good patch do we need for a non-trivial solution to this problem? And so this is the picture here. So this is the upstream side, this is the downstream side. The flow is moving to the right. And we have boundary conditions that relate the density to the slope on the upstream side and the downstream side, and they're different. And so this would be a possible non-trivial solution to the problem at the top with the boundary conditions. And it turns out it's possible to compute the solution to this problem exactly. And it's kind of a nice problem, sort of critical patch size problem with slightly exotic boundary conditions. And so that would be something that mathematical ecology graduate course or undergraduate course, upper level undergraduate course might do. And so one can work out the critical patch size here. And here it is. And just to make it sort of interesting, I've put the C star in here, the intrinsic spreading speed, which is 2 times the square root of R times D. So there's a formula here. And here we see the intrinsic spreading speed definition. But notice what happens now as the advection speed approaches the intrinsic spreading speed. This denominator blows up, okay, because we have 1 minus 1, and this also blows up. So, so when we have L below L star, there's just a trivial solution. And when L exceeds L star, then we have a non-trivial solution. So we can think about this as a transcritical bifurcation, where here we're tracking the maximum height of this steady state solution. And so if we put this on the graph that we saw previously, now we have a new axis, and that's the critical domain size L, which is shown right here in dashed. And so the critical domain size looks like this. It grows with the advection speed. And then exactly at the point where the advection speed is equal to the intrinsic spreading speed, it becomes infinite. And so this actually is a nice way to connect these two concepts of spreading speeds and critical domain size problems through this fact that we get the blow up exactly at the stalling point for the upstream spread. And it's nice to teach this because it allows you to bring these two areas of kind of beautiful mathematical work that has a lot of history together in this way where they're somehow connected. Okay, so this is going to provide a basis for looking at population persistence during range shift. And so we want to go back to what we're talking about originally, and that this is the case where there's a climate envelope that's shifting at speed c to the right. And we'll just stay in one dimension for now. And the size of the climate envelope now will be L. And so we incorporate the shifting climate envelope into the reaction diffusion model by actually letting it move to the right at speed c. And our model otherwise looks just like the previous model, where we have growth inside the patch and mortality outside the patch. And again, this is a reminder of what the different parameters are in the model, and they have not changed. The only new thing is this C now is being used to describe the speed of the climate change, the rate at which these isoclines are moving northward. And so it's possible then to take this model and just shift to a moving coordinate system. So into the system x minus ct is psi. And then we have the following equation shown here. And then this is very much like our previous model, and it becomes identical if we let A be minus C. And so, so that allows us to use that theory that we just talked about now in the climate change scenario. And so there's a theorem about this model, and it's fairly straightforward. It says whenever the intrinsic spreading speed of the population exceeds the rate at which the isoclines are moving, there exists a critical length, which has got the familiar formula we saw before, such that if the length of the patch is less than the critical length, the population will go to zero uniformly. And so this is the picture here, is that first of all, we require C star to be bigger than C. So if you can't keep up with the climate change, then you're not going to survive at all. But if C star is bigger than C, then you need a critical length. And if you fall below that critical length, then your population goes extinct. And so you might ask, what happens if you're above that critical length? Well, when L exceeds the critical length, there exists a traveling pulse solution that moves at the rate of climate change. And so it just follows this traveling pulse solution. And reasonable initial data will converge to this pulse for a large time. And so this is work I started with a postdoc or research associate, Alex Potapov, in 2000, and then Veristicky and Rossi and some other researchers, Odo Diekman, looked at this and made the mathematical results much better and kind of extended the work. And so, so here's a traveling pulse, and it's moving to the right now at speed c. And if the astute observer will see, I have also sliced this like a loaf of bread and have these different neutral genetic fractions. And so if we look at this diagram, C star versus L, this particular pulse is in the survival region. And so we know that this is going to be a reasonable solution that will exist. Okay, so now we want to ask the same question as we asked earlier. What's going to happen to these neutral fractions as this pulse moves northward? And so we basically take the approach of Garnier and Rock, and actually this work was done by Jimmy Garnier when he was a postdoc with me a few years ago, and so the approach is very similar to the earlier work that was for the unconstrained expansion into a novel habitat. And so again, we break the population down into these neutral fractions, and we ask how they change over time as the front moves forward. And so it's a very similar question, but now it's for this forced pulse that's being driven by climate change. And again, let's look at the computer simulations first and then try to make sense of them mathematically so we can gain some intuition. And so the unconstrained expansion scenario is shown in panel B, and it's very much like the previous results, where it's just the population at the leading edge, which is driving the expansion. And so we lose genetic diversity in the leading edge of the wave. Now we have a case where we have a shifting habitat, and it's shifting at speeds c is equal to 2. And asymptotically, we see a variety of different colors. We see more gray than red and more red than orange and so forth in this case here. when it's shifting its speed too. And then when it's not shifting at all, it's just a stationary patch, then we see that after some period of time, the population seems to have all the different colors more or less equally. And the founder effect is most pronounced with this unconstrained expansion, and it's least pronounced from these numerical simulations with stationary range boundaries. And when there are range boundaries, it appears that the densities asymptotically approach the same profile as the wave. And so each of these different colors appear to have the same density profile as the total density as time goes to infinity. And it turns out that the numerics show that faster moving range boundaries give a strong founder effect. And so when the range shift is fast, you see more gray than when it's not so fast. And I've just repeated those points again at the top of this slide. And it turns out there's I shouldn't really call this a theorem. It's more like a corollary of the earlier work. We can reapply the methods that Garnier and Rock used to look at population spread. And there's a very similar result that pertains. And this result is like the result that was for the Lie dynamics. And so for any initial condition, the density fraction for the neutral fraction converges uniformly to a proportion of the total population density. And so what we're seeing with those numerics is actually possible to prove that asymptotically, each of the different neutral fractions will achieve the same profile as the traveling pulse. And there'll just be a fraction of that traveling pulse, P, and P will depend on the initial distribution, V naught. And the formula for P is like it was before. And so here, this is the initial fraction of the initial distribution of the neutral fraction. This is the traveling pulse, and this is a weighting function that weights things near the leading edge of the wave more than things at the back of the wave. And I forgot to mention earlier, but this term on the bottom is just a normalization term to make sure all the P's add up to 1. And so we can see as the climate change speed increases, greater weight is given to fractions at the leading edge, because this term e to the c over d is going to be larger if c is larger. And so that weighting is going to be stronger. And so this again is just a reminder of what we saw. So in this case here, the weighting provides more advantage to the gray than the red, and so on and so forth. Whereas here with c is equal to 0, that weighting term in the formula would disappear entirely. Okay, so there comes a point where you sort of work on things mathematically and you get some interesting results, and then you're thinking about how am I going to communicate these maybe to people who don't love analyzing partial differential equations and try to make an impact in biology. And so Jimmy and I thought about this, and so we wanted to measure for diversity during range shifts that we could talk about. And we know there's a formula for the asymptotic proportion of any neutral fraction, which was on the previous slide. It's P of V naught of I. And if you have a whole bunch of neutral genetic fractions, you can compute the genetic diversities. And then you can define something called asymptotic diversity of order Q, which is given here. And it turns out that Q controls the emphasis based on common and rare fractions. And in the limit, as Q goes to 1, the asymptotic diversity is closely related to, it's like the exponential of entropy. And so it's some measure of, classical diversity. And so as Q goes to infinity, the focus is just on the most dominant of the fractions. And so what we can do now is we can just plot that asymptotic diversity function. and then say, how does that change as we change interesting quantities? And in our case here, on the left, we're taking asymptotic diversity of order 2, on the left 2, and then asymptotic diversity of order infinity on the right 2. And the main point here is that it doesn't actually matter what index you use. We've tried a bunch of them. They all show the same pattern. And so somehow the results are qualitatively independent of which diversity measure you're choosing. And then on the horizontal axis is the spreading speed. And we change the spreading speed by changing the diffusion coefficient D. And that's what's shown here. And on the vertical axis is the size of the climate envelope. And so if the spreading speed is too small, then we're in a region here where the population diversity is 0 because the population doesn't persist. And again, if the climate envelope is too small for some given spreading speed, then the population won't persist either. So this solid line here shows the demarcation between persistence and extinction. And then if we're in this region that does persist, then the color shows the diversity, and so, or the asymptotic diversity. And so we see a red ridge here of high diversity, and then as the colors get cooler, the diversity is decreasing. And so we can compare, say, the first row with the second row. And the second row shows the situation for fast climate change or faster climate change, whereas the first row would be slower. And it's possible to show that the faster climate change is eroding the diversity. Okay, so the deep red colors aren't seen in the bottom as much as in the top. So we see that, but we also see that if we fix the intrinsic spreading speed, C star, at some value, say 15, and then we think about increasing the envelope size, we get it to a size where the population persists. And then initially there's low diversity. And then as we get larger, the diversity increases. And then as we get even larger, the diversity decreases. And so somehow a very large envelope is going to lead to decreased diversity, and a very small envelope is going to lead to decreased diversity. But an intermediate size envelope is going to be the best in terms of diversity. And we can do the same thing if we fix the size of the envelope and then change the spreading speed for the population. When the spreading speed is too low, it falls below the climate change speed, and so there's no persistence. And then initially is low diversity, and then at intermediate spreading speeds there's higher diversity, and at very high spreading speeds it's lost again. And so we think of high spreading speed leading to loss somehow at the front of the envelope, and the low spreading speed leading to loss at the back of the envelope. And so if we put all this together, just using reaction diffusion equations, we've kind of theoretically assessed the impact of range shifts on neutral genetic diversity of a population. And why I like talking about this is that I think it ties in with a lot of things that we study in mathematical biology in terms of even undergraduate discussions of population spread and critical domain size problems. And another reason I really am excited about this is because of these recent experiments by Gandhi et al., where they've tested some of these ideas on yeast populations. But the mathematical theory that I've been drawing on here and talking about was introduced by Rock et al. About 8 years ago. And this is the idea of using these inside dynamics to see how population diversity changes over time. And then, so we've extended this to include these heterogeneous pulses that would come from climate change. And then kind of extended it also to look at how we can summarize the results using diversity indices. And kind of just a little recap. So the stationary climate envelopes would sort of, from our theory, would promote the highest levels of diversity. And but when they move, then the diversity, like Goldilocks didn't want her porridge too hot or too cold, just wanted it at the right temperature. And it's a little bit like that with both the climate envelope size and the intrinsic spreading speed. So for higher diversity, the climate envelope size should not be too large or too small. And the intrinsic spreading speed should not be too large or too small compared to C. And I just wanted to give a shout out to a recently finished graduate student, Nathan Markoulis, who has done a lot of work extending these models to different formats, and in particular integral difference equations that can be more closely related to the biology. And he's also done things like he's included mutations and so forth in the structure there, as well as fat-tailed dispersal kernels and so forth. So I guess I just want to say thank you. I had a lot of colleagues that have given me a lot of feedback, and I acknowledge funding. And I do have references at the end of my talk, which if anyone's interested in, I can make the talk available, and then people can look up those references or email me if they want.
Thank you very much, Mark, for a very insightful talk. So the floor is open now for questions. Anybody? There's a question that is actually presented at the chat room from Marina. Would you like to present your question, Marina, or shall I read it?
No, I can say it. Thank you very much,
Sure.
Dr. Kadra. Hi, Dr. Lewis.
Hi.
We've met before in Jasper, so very nice to listen to your lecture again. And very interesting. Thank you very much. Question: with the changing isocline, do we expect the diffusion coefficient highly dependent on the space dimensions?
Okay. Yeah. So I guess the work I presented today is just in one dimension, and the same process in two dimensions would have diffusion coefficient that would scale to 2 dimensions, and there's a possibility of much more complicated structures in 2 dimensions because of the shape of the patch. Jimmy Garnier and I have done some, mainly just Jimmy, has done some simulations of these same kind of processes in 2 spatial dimensions. And you can get qualitatively different results depending on the shape of the patch. So you could think of a circular patch versus a very long rectangular patch and so forth. And these play a role, and the orientation of the patch shape relative to the climate change also plays a role. So I think it's a very rich area for further research, and we've
yeah, I wasn't clear enough. I'm sorry, I apologize. I meant in diffusion reaction equation, we take out D as a constant in the case it's independent of X.
Oh,
but
yeah.
we are changing isoclines, and it goes in some, a really different area for this species. Should we, so there's going to be a really interesting solutions in this case.
Oh, I think I understand. Sorry, I kind of misunderstood. But I think if I can just reinterpret it and tell me if I'm interpreting it right, you're thinking that maybe the animals or the populations would disperse differently in different kinds of habitat.
Yeah.
And so there'd be a D that would depend on whether you're inside your patch or outside your patch, for example.
Yeah.
Is that correct?
Close enough.
Okay. Yeah.
Very quickly.
So yeah. So I think I mean, Fritjof Lütcher has worked on this very problem with one of his grad students, where the movement term depends on the location in the habitat.
Yeah.
And of course, there could be a tendency of animals to prefer habitats that are better for their fitness, and so you can include a bias in the movement term to allow you to go back into the good habitat. And so this is definitely an interesting extension.
Very quickly, the second question, if I may. Thank you so much for this interesting including of Ali effect. I just did some work on Ali effect, and it was very nice to see how it affects the neutral fractions. So I was wondering, is there some works available for the mixture situation in terms of mixed pushed and pulled cases?
Oh, yeah. So you mean at the transition between pushed and pulled?
Yes,
Is that
yes,
what you're
because
thinking?
it's
Yeah,
it's
yeah,
sort of more natural situation, doesn't it?
yeah. That's right. I think I have to check here. I think the results by Rock et al. Might also apply to the weak-alley effect situation. So the weak-alley effect, I'll just go back a ways here, is the one that describes a population that looks like this here, where there's the per capita growth rate is diminished at low densities, but it's not negative. And a strong alley effect would be a curve that would come down and go back up again. And I'm trying to remember if I should check this actually. I actually, yeah. So do they assume that the alley effect term is A is greater than 0? I don't know. Yeah, so I'd have to check that. Yeah.
Yeah, that's okay. Thank you so
Yeah,
much.
that's a very good point. I should check that. Yeah. 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 Thank you.
Any other question? Okay. Go ahead, Grigor. Go ahead.
Hi, Mark. Very nice.
Hi. Thanks.
I was interested in this Goldilocks phenomenon that in the intermediate ranges in the envelope you have the maximum diversity, and I didn't quite catch how the model is set up. What happens once you are in the envelope? Is your population growth rate constant, or is it still depending on how bad or how good the environment is, gradually better or higher or lower? So is that the reason for the Goldilocks phenomenon? If you think temperature, there's a right temperature, but if it's too high or too low, your growth rate is lower than the optimum, or is that a different effect?
Yeah, so I think it's a different effect, but in the model that I presented today, within the good habitat patch, it's just simply logistic growth. And outside of that habitat patch, it's exponential decay. Okay. So just density independent mortality. And there's no there's a abrupt transition from one to the other as you go through the edge of the habitat. I think one way I understand the diversity effect is if you have a very small habitat patch, the population is just on the edge of disappearing, and those fractions that started near the edge of the habitat are going to wash out and be lost. And so you'll only get the individuals that were initially really close to the middle of the habitat persisting and giving rise to the diversity in the future. And so that's the case where the habitat patch is small. When the habitat patch is large, you have something which is a little bit like the founder effect, because if you have an infinite habitat patch size, then as the population spreads, it'll just be those individuals at the leading edge that give rise to the new genetic composition of the population. And so it's sort of this trade-off between the kind of classic founder effect and then the loss through the boundaries that drives the Goldilocks effect. When you have an intermediate size patch, if you think of a population growing and dispersing, and that patch is moving, there is at least an intuitive connection to the case with an alley effect, because what happens is at the middle of the patch, populations are growing quicker, and at the leading edge of the patch, individuals are dispersing out and dying, and those individuals in the middle of the patch are then spilling over and filling in the gap left by those at the edge. And so somehow these intermediate sized patches have a dynamical property which is somewhat similar to the Allee effect, but of course it comes from a very different origin.
Okay, good. Just so that I'm clear, when you say habitat patch size or patch size, habitat size in the climate change case, that's the distance between the two isoclines where the population still has positive growth rate. Is that correct?
That's right, yeah. Okay. Yeah, so that's exactly right. So, and of course, with that in mind, it's rather simplistic because it's possible that the front and the back isoclines would move at different speeds.
Okay, yeah.
Yeah, which is another interesting area for mathematical analysis.
Okay, thanks.
Sure.
Okay, any other question? Okay, maybe I'll ask mine since we're waiting for the other people. So I have few questions. When you talked about the climate change example that you had and the push versus pull models, and now if I got it correctly, the push one is the one that have low genetic diversity because the only one that is spreading, this fractional component, only one component that is spreading, right?
That would be the pulled wave.
The pull, sorry, the pull
yeah,
one.
The pulled, yeah,
okay.
Yeah.
So the question that I have is that why would then the population spread if you have a decrease? I mean, I know it's coming from the mathematical model, but wouldn't that be counterintuitive? Because my understanding is that the lower the genetic diversity, the less resistant the population is to climate change.
Yeah, okay, so you bring up a very good point, and that is that in the models I talked about today, the neutral assumption is made. And so the idea is that all the gene fractions have exactly the same fitness. And so what we're looking at is the spatial redistribution of these fractions through the dynamical processes associated with growth and dispersal. Of course, if you confer different genetic advantages or disadvantages to gene types, then the story changes a lot. And there is some work on this kind of stuff, and it's very nice, using sort of quantitative genetics models where there's some diversity of genotype, and then there's also distribution in space and time. And that turns out to be a different story because then you could allow populations to evolve as well as genetically mix. Yeah, so this is most akin to in a population if you have like some of these single nucleotide polymorphisms that are markers on populations that are used to assess genetic diversity, and you'd want to know how they change, where there's no known selective advantage to having that particular polymorphism.
Oh, okay. Okay.
And so in a sense, it's a very specialized case, and so I kind of think of it more like a base case,
yeah,
right? That we get some kinds of insights from, but it's still experimentally replicable, like with those yeast studies, right? The mathematical theory was actually confirmed with the experiments with and without the Allee effect, but it's somewhat it's not the most general case.
So can we, since you're mentioning it, you're coming to my second question. So when you look at this experiments you presented with the yeast, sucrose versus glucose, can you actually replicate some of these climate change experiments by starving these cells, like somehow subject them to starvation periods where you could see how that genetic diversity is affected as a result?
Yeah, well, I'd love that to happen. Yeah, I wrote commentaries on the two papers. Both of these papers were by Gandhi et al. They were published in Proceedings of the National Academy of Sciences, and I wrote commentaries of each of them, and I was very excited about them. So I think maybe I'll just have to write to the authors and ask them if they will do one more set of experiments.
Okay.
And definitely very cool stuff. It's nice to have a experimental setup where you can test theory, right?
I think it's doable.
It's so rare. Yeah, I think it might be. Yeah, it would have to be a little bit ingenious getting the shifting habitat, right? But I feel like there could be a way.
Yeah. Okay. So any other questions, folks? Last call. Okay. So thank you very much, Mark, for I guess I'm speaking for everyone. It was a great talk. We really enjoyed it, and thanks for participating in this seminar series.
Great. It was a great pleasure. Thank you for having me.
Take care.